Your evening chocolate treat may sabotage sleep. Caffeine, inside chocolate, blocks adenosine receptors in your brain's basal forebrain. This receptor blockade occurs as soon as 30 minutes after consumption, delaying your brain's sleep signal. Adenosine, a byproduct of ATP metabolism, builds up during your day, signaling sleepiness. But caffeine's interference disrupts that accumulation. Our need to sleep doesn't stem from exhaustion alone. Instead, it's a calculated process involving adenosine accumulation and receptor interaction. Understanding this intricacy debunks the myth that a small chocolate indulgence is harmless. Instead, even minor caffeine doses alter sleep readiness. Eating late at night disrupts your body's core temperature rhythm, a crucial part of circadian biology. The SCN, or suprachiasmatic nucleus, regulates this rhythm, peaking in the early evening. Late-night meals delay this peak, shifting the SCN's phase and disrupting your sleep onset. A study by the Salk Institute showed that eating 3 hours before bed increases wakefulness. This time delay impacts melatonin release, a hormone your pineal gland secretes to signal sleep. Thus, late meals aren't just about digestion, they shift circadian timing, altering sleep initiation. Alcohol before bed might seem to promote sleep, but it disrupts sleep architecture. Alcohol reduces REM sleep by increasing GABAergic activity in your brain. This neurotransmitter inhibition elongates NREM stages at REM's expense. Research in the Journal of Clinical Sleep Medicine found reduced REM sleep as much as 9% in moderate drinkers. REM, critical for memory consolidation, is compromised, affecting cognitive function. Thus, alcohol's sedative effect belies its underlying disruption of critical sleep stages, revealing the deception of its apparent sleep aid. Sugary snacks spike blood glucose, affecting sleep by altering orexin release. Orexins, produced in your hypothalamus, regulate arousal and wakefulness. A high-sugar snack, measured in a 2019 Stanford study, reduces orexin activity, leading to excessive daytime sleepiness. High glucose levels cause insulin spikes, which subsequently suppress orexin. This suppression leads to a cycle of tiredness despite adequate sleep duration. Orexin's role in maintaining alertness is thus compromised, demonstrating how sugar impacts energy regulation beyond simple caloric intake. Even seemingly innocuous foods like aged cheese can affect sleep through tyramine content. Tyramine prompts norepinephrine release, increasing alertness. Aged cheese boosts synaptic norepinephrine levels, studied by the University of Chicago in 2020, raising sleep latency. This neurotransmitter spike causes heightened brain activity, similar to stress responses. Such activation can delay sleep onset by up to an hour. Thus, foods rich in tyramine, like cheeses or cured meats, can silently extend your waking hours, affecting your natural sleep cycle. Inside your neurons, adenosine accumulates, creating sleep pressure. This compound, a byproduct of ATP breakdown, binds to A1 receptors, inducing drowsiness. However, caffeine molecules mimic adenosine, competing for these receptors. This competitive inhibition prevents adenosine from signaling sleepiness. Normally, adenosine levels peak in the evening. But caffeine's presence blocks receptor binding, delaying sleep onset. The interaction occurs predominantly within your brain's basal forebrain. This precise mechanism shows how caffeine's structure disrupts natural sleep signals, extending wakefulness artificially. Your brain's glymphatic system clears metabolic waste during sleep. Astrocytes, or glial cells, facilitate this clearance via aquaporin channels, expanding interstitial space. This expansion accelerates cerebrospinal fluid flow, removing amyloid beta and tau proteins. A 2013 study from the University of Rochester demonstrated that glymphatic activity increases by 60% during sleep. Alcohol and other depressants inhibit this clearance, impairing cognitive function over time. Hence, substances affecting sleep stages also impact waste clearance, revealing a critical link between sleep and neural health. Circadian oscillator genes, like clock and BMAL1, synchronize your body's internal clock. These genes regulate transcription factors, modulating brain and liver functions. High-fat diets alter these gene expressions, delaying circadian rhythms. A 2016 study in Nature Communications found altered oscillation phases in mice fed high-fat diets. This delay shifts metabolic processes, impacting sleep timing. The interplay between diet and gene expression underscores how specific foods affect more than caloric intake, extending to genetic regulation of sleep cycles. Cortisol, known as the stress hormone, peaks in the morning. This cortisol awakening response aligns with your circadian rhythm, promoting alertness. High-sugar diets can alter cortisol peaks, augmenting stress responses. A 2018 study at Cambridge University found increased cortisol levels in individuals with high sugar intake. Cortisol's interaction with sleep involves its regulation of the HPA axis, affecting REM and NREM balance. Thus, diet-induced cortisol changes can disrupt sleep architecture, demonstrating how biochemical responses to food impact sleep quality. REM stage sleep consolidates memory by processing recent experiences. During REM, the hippocampus communicates with the neocortex, enhancing memory storage. Alcohol reduces REM frequency, as shown in a 2017 meta-analysis of sleep studies. This reduction impairs memory consolidation, highlighting REM's role in cognitive function. Orexin signaling, affected by diet, further modulates REM duration. Suppressing orexin through high glucose intake decreases REM stages. This intricate interplay between neurotransmitters, diet, and sleep architecture reveals the multifaceted impact of late-night food choices on cognitive health. Chronic sleep debt alters metabolic regulation, increasing the risk of obesity. Sleep deprivation affects leptin and ghrelin, hormones regulating hunger and satiety. A 2004 study from the University of Chicago found that sleep restriction decreases leptin by 18% and increases ghrelin by 28%. This hormonal imbalance leads to increased appetite and cravings for high-calorie foods. Over time, sleep debt contributes to weight gain, emphasizing the intricate link between sleep and metabolism. The long-term consequences extend beyond immediate fatigue, affecting your body's energy regulation and appetite control mechanisms. Acute sleep deprivation impacts cognitive performance, affecting decision-making and attention. In a controlled study by the Walter Reed Army Institute, individuals deprived of sleep for 24 hours had reaction times 50% slower. This delay parallels alcohol impairment, underscoring sleep's role in cognitive function. Foods high in refined sugars exacerbate this effect, spiking blood glucose and resulting in energy crashes. These crashes impair cognitive processes further, illustrating how diet and sleep deprivation combine to degrade cognitive performance. Chronic deprivation compounds these effects, impacting brain health over time. Sleep tracking apps often misrepresent true sleep quality, focusing on duration over architecture. Polysomnography, the gold standard, measures sleep stages, revealing disruptions not captured by consumer devices. For instance, a study published in Sleep in 2020 showed that sleep trackers overestimate sleep efficiency by up to 20%. This discrepancy can mislead users about their true sleep health. Sleep architecture, including REM and NREM balance, is crucial for recovery. Thus, reliance on simplistic metrics overlooks the complex nature of sleep, potentially masking underlying issues. The wellness industry often promotes supplements like melatonin without acknowledging variability and effectiveness. Peer-reviewed literature reveals that lifestyle, including diet, significantly impacts melatonin efficacy. A 2019 meta-analysis found inconsistent results, with effective doses varying widely among individuals. Dietary factors, such as late caffeine or alcohol intake, reduce melatonin effectiveness by altering sleep onset and duration. These interactions challenge the notion that supplements alone can rectify sleep issues, emphasizing the need for a comprehensive approach that considers diet's role in sleep regulation. One study from the Journal of Clinical Sleep Medicine in 2018 revealed that those consuming higher-fat diets had a 45% increase in sleep disruptions compared to low-fat diets. This fact reframes the idea that sleep quality is merely about hours logged. Instead, nutrient composition plays a significant role. High-fat intake alters sleep architecture, impacting deep sleep stages necessary for recovery. This finding challenges assumptions about calorie content, illustrating how macronutrient balance influences sleep health. Understanding this connection highlights the importance of dietary choices beyond mere energy intake.